kindly assemble in the audience. We are about to start with the keynote session. Coordinators, please look for the participants and the students. Coordinators, please kindly ask everybody outside the auditorium to gather in the auditorium fast. And students who are sitting quite a little back, particularly the students sitting in the middle aisle, sit in front, occupy the front rows of the seats. Kindly please. I request all the students, not only the middle aisle student, all the students come and sit in the front because we are live on the YouTube streaming. So kindly get up and sit in front rows. आप सभी से निवेदन है कि आप आगे आके बैठ जाएं.
Okay. Hello. Should I come?
Yeah, I introduce you, sir.
Okay. I will stay here.
With due permission, I would like to begin with the keynote session 1. I take this opportunity to introduce our keynote speaker of the day, Dr. Varun Kushwaha sir. Sir is Senior Scientist at Central Drug Research Institutes, CDRI, Lucknow. Prior to joining CDRI, he served as a Principal Research Scientist for over 6.5 years at the Research Center for Pharmaceutical Engineering, Graz, Austria, and worked as a formulation research scientist at Pfizer, Chennai, India during 2017-18. He earned his PhD in 2017-18 from the Center for Pharmaceutical Nanotechnology Group at the National Institute of Pharmaceutical Education and Research, SAS Nagar, Mohali. Dr. Kushwaha has received several prestigious honors, including the Fulbright Fellowship, the Commonwealth Scholarship, and the SCI Scotland Best PhD Award. His research interests include nano drug delivery platform, bioconjugate system, and advanced solid oral formulation. His current work also involves developing predictive model for his in vitro release profile of biodegradable, long-acting injectables, aiming to accelerate their market readiness. To do the honors, to welcome sir, I will request Dr. Ashish Chaurasia sir to welcome sir with a sapling. Please, sir. Please welcome our keynote speaker.
Thank you. Yeah.
Thank you so much. Now I would ask Varun sir to take over the session. Over to you, sir.
Okay. Hello. Yeah. First of all, thank you very much for inviting me and giving me an opportunity to interact with the young leaders of India. Can you hear me in the backside? Okay. There is a one deal. If you don't ask me question, I will ask questions. Okay. So be cautious. If you want, you can come in front. So kindly allow me to introduce myself. Already introduced very well from the colleague. Myself, Dr. Varun. I am a senior scientist in CDRI Lucknow. Recently joined here, and almost I have a 7-year experience in industrial research, hardcore research institute in Austria. To be honest, I am here to interact with you guys, and the slides and the presentation is mainly dedicated or prepared in focus to you guys. The whole presentation is prepared like that you will get the most benefit out of this. So believe me, if you are tentative for half an hour, it might change your whole life. So I think it is one of the best opportunity if you are in this phase of your life. So if you just give some attention to the slides and what I am saying, okay? So mainly three type of students will get benefit of this presentation. The one type of student is that who wants to pursue his or her career in research, okay? How to proceed or how to do good in research field. The other thing is that if you want to go abroad, outside India, for your academics, for your professional life, then this presentation is also dedicated to you, okay? And the third and the most important thing is, if you want to be an entrepreneur, okay? If you want to start your own journey after your academics, for example, after master's or PhD, then this is also one of the best opportunity for you guys to understand how to move forward. So the three things is one for research scholar, the other one is who wants to pursue his or her career in outside India, and the third one is who wants to start his or her own journey as an entrepreneur or as a startup organization. So I will go slowly one by one. If there is any questions, please feel free to ask me. Because it will also give me some understanding that if you are getting a point or not. I am again saying that please feel free to ask me anything because it is most critical for you, because for us it's just a matter of giving a presentation, but for you guys it's just one really big opportunity. Not only for me, also with Arvind and Isha, if you get some questions, if you have some questions, please feel to ask when you are comfortable. And let's start the journey of slide, okay? So you might have known that this is just academic blocks of CDRI, where I am currently doing my senior scientist job there in pharmaceutics and pharmacokinetics division. I am mainly a formulation guy, like who makes formulation or drug delivery systems for any API, mostly for anti-cancer drugs. I am mainly dedicated. Yeah. So this is my journey. The orange color shows you my industrial experience, and I think this is like slightly of green or turquoise color. So the turquoise color is showing my academic career, okay? And in this box is the list of companies which I worked with, okay, during my 7 or 8 years of experience. And you can see that I started my bachelor's from Dr. Hari Singh Rao University, Sagar, and then I moved to IIT BHU for my master's, and after my master's, I went into CSIR Mohali, Naipa Mohali, I think scoring or getting rank in CSIR net. So I qualified that too, so that I am guaranteeing myself that I will get a PhD after my master's. Within my PhD, I went into UK under the Commonwealth for split side PhD. So the basic thing is it's in your PhD. PhD is almost 4 to 5 years of duration. So in 4 to 5 years of duration, on the basis of your academic skills, you get 1 year or 2 years of your time where you can actually continue your research outside India, mainly in UK. So Commonwealth is an organization who gives financial funding to you, not only to you, for your also research work. So you will get some monthly around 2 lakhs per month of scholarship, which is free only for you to, for your expenses, and also gives really a huge amount of money for your research. So it's one of the best opportunity for any researcher to go there. So you can just Google it, this Commonwealth scholarship. There is a dedicated website, so you can just Google it and then apply for any streams. So there is master's stream also there, PhD streams are also there, or split side PhD is also there. So depending upon your needs, there is a different website, or there is different links where you can do your research in UK. So UK means Scotland and London, or UK, or sorry, Great Britain, you can say. After completing my split side PhD in UK, I went to US in University of Louisville, Kentucky. It's also similar to Commonwealth Fellowship. There is a Fulbright scholarship, which is similar to the Commonwealth, but it is dedicated to do research in US. Any research organization in US, for example, Boston, MIT, you name it, and they are giving you the opportunity to do your research there. So it is also possible that if most of the people I think are from bachelor, so you can also do masters from Boston, for example, or MIT if you get qualified to Fulbright. So there is also a dedicated website for Fulbright, and you can just Google it, search it, and if you get this opportunity, I will say that this is one of the best opportunities, because it's a elite group of researchers. And normally, I think 14, 50 or 30 students are selected each year to do research in US, okay? The best thing for both the scholarships is that after your academics, you get 2 or 5 years of work visa. So there is no need to come back, after doing research or after completing your degree, you have to come back to India. No, it's not like that. You get dedicated research work permit, so you can continue your work in industry in US or UK or wherever you want to do. So I think after completing my scholarship, Fulbright Nehru scholarship, I came back to India, worked in Pfizer as a research scientist, and after that I also went into US Pfizer to Boston to work with Pfizer there. And after Pfizer, I went into because in my research career I have seen UK, US, but I was just a little bit curious that how Europe works. So that's why I choose Austria to do some science, or as an industry I wanted to see how the industry works. So I joined there, and I almost worked 7 years. And during these 7 years, you can see this list. It's not the complete list, but just some, so that you can get some feedback how the industry works there, or how they are collaborating. So you can see this Pfizer, USA, J&J, Belgium, AstraZeneca, UK, KC, Sandoz, GSK, Syngene, BI, Debut, Gentiva, CMAC, Merck, AMS, Anton, Paragus, Belform. There are n number of companies which are actually curious still nowadays to do research and collaboration with global, collaborators. Even now, because I know very precisely, and because I have pretty much of good interaction with all these companies, they are even now interested to come to India to collaborate, because the number one is the cost of the resources. The resources means labor, like a scientist. The cost of scientist is very low as compared to European or US countries. So that's one opportunity there. And if you are getting some opportunity, if you are interacting, maybe you can use LinkedIn platform to interact with the guys. If you get some opportunity, they will definitely entertain you, and they can also fund you for your research. And if they are happy with your research activities, they can also hire you upfront. They will upfrontly say that, okay, you are nice, doing your nice work, and please come to our site and just to do research there. So there is a huge possibility of getting a job or research project there if you want to do like that. So there are multiple streams where you can collaborate and work in outside India. So this is one part, okay. And after my 7 years of research activities in Austria, I came back to India, and I joined as a senior scientist in CDRI Lucknow. And there might be some questions that why you want to come back to India. So first of all, that I want to be in India because it's my motherland. And the second thing is I want to give something back to the pharmaceutical society in India. And also started a small company, which is Varisha Life Science Private Limited in IIT Kanpur. So that's, as I told you, the last part will be entrepreneurship. And trust me, I have seen each and every country, mostly all the 27 countries of Syngene, US, UK, Scotland, whatever you say, I have visited it. But if you are an entrepreneur, that is really give you all the best piece of pleasure and freedom, because you can do whatever you want and whatever you have desire to do. But before that entrepreneurship, you have to take some experience, go abroad, go do really good science, not only just simply write a paper, false results. It doesn't really help you out. So just to do research, to have a passionate towards research, and then go abroad, do some research there, get some experience, come back. There are multiple opportunities, for example, IITs, NITs, NIPers, who are giving you really a very good platform for doing research there, and as you starting your own startup. So I will now move to I will show some of the examples where I can show that, okay, how you have to understand how to do a research, how to pick the topics, that will be really good for you to understand, okay, how to do good science and really meet the standards of global scientific things in world. Yeah, so that I was awarded by Fulbright, Commonwealth, and for my PhD I also get the best PhD award there. I qualified all the entrance examinations in India, for example, CSIR NET, GPAD, GATE, whatever you say. I have actually done this. I got also a travel grant, and I have 86 international research papers, book chapters, conference, patents, and also guest editor and associate editor of one of the international journals. So you can just Google it out and you can have a look at it. I have 3,193 citations with an H index of 38 and I index of 64. So once you get into this stream of research, the caliber of your research is actually evaluated from these things: citations, H index, and I10, and also the number of papers you have as a first author, as a corresponding author. So first author, that, okay, the first name of in the paper, right? But the corresponding author is that if you have a lab and you are giving your facilities and supervising a whole research work, then you will get a corresponding authorship, which is the last name of the list of authors in the research paper. And during my 10 or 7 years of research in Austria, I have completed more than 50 research projects. With all the 50 projects are not academics; they are all industrial research, peer research, which is worth more than 5 million euros. So that's why I'm saying that if you have some knowledge about how the people are doing their research there in all globally, then you can really, believe me, you can do your own startup very easily in India, and the people will come to you, that the people will like to collaborate with you. So I will give you some of the examples. One is the case study of Ojodex. So this is Ojodex is an implant, small implant, which is, you can see, it's 6 millimeter in length and 0.5 millimeter of diameter. So you can understand that it is really very thin. And another one is nanoparticles, because I have always heard from the people that nanoparticle is just a bluff. Okay, there is nothing like nanoparticles. It's not industrial practical and all. So I will say that really there is a huge potential on it. And I think if you focus your research mainly on this implants, medical devices, and nanoparticles or advanced drug formulations, then definitely you will get a really good profit out of this for your professional life. So I will go with for case study 1. So you can see this Ojodex is a dexamethasone intravitreal implant, which is very small and is normally injected into eye. You can see here. And here it is injected by surgical process. But the problem is, there are 3 major problems about that. One thing is the cost. Okay, so the cost is, you can see, 1,400 euros, so which is equivalent to 1,20,000 rupees in India. That is one of the reasons that most of the patient cannot afford it, and their vision is not prolonged because of the diabetes. It's mainly used for diabetic retinopathy. So as you know, it is developed in US. That's why we have to pay that much of amount to protect our eyes, but that's not possible. The second thing is, there is no generic, because the implant is prepared from biodegradable polymer, which is PLGA, and PLGA, there is very less number of research paper done on this, research work is done on this, and as a result of it, there is no generic till now in market. So whatever, if the patient is suffering from diabetic retinopathy, he have to pay over 1 lakh for one dose. Okay, and even after paying 1 lakh of rupees for one dose, what you get, the quality of the marketed formulation is not that quite adequate. As you can see in the second picture, within the eyes, after a few months, 1 month, it is actually broken down into 2 pieces, and no one knows what happens now, because you cannot take it out from the eyes, right? So just imagine, you have one operation to inject the eyes, inject the implant into the eyes, and the second operation to take out from the eye. So it's really a big drawback for that. So there are a few things which we wanted to focus on. That first of all, we want to make a generic formulation, and not only for this, but can we do it as a platform technique? So even though in future 10 years, if some new formulation comes, that we have already pre-planned this, so that there will be a generic or an innovator formulation even beforehand, right? So you don't have to pay that much of amount, because the total cost for developing one formulation is around 5,000 rupees only. So just see, 5,000 rupees and then 1 lakh rupees. So you have to pay because you are not the innovator. So India is not an innovator, so you have to pay for it. So the implant is mainly made up of PLG, which I mentioned to you, 502H and 502. It's a mixture of two polymers. One is H means hydrophilic, and 502 is hydrophobic in nature. So this is the chemistry, and you can see in the 502H, because of the presence of COH group, which is carboxylic group, it has high affinity towards water molecules, and as a result of it, it's more hydrophilic in nature. But in the case of 502, because you can see there is a CH3 group here, CH3 group in the outside, in the left side. I don't know. You can see. I think you cannot see the pointer, right? So that's why, because of the presence of CH3 group, it doesn't like water, and it's a hydrophobic in nature. And the implant is made up of in mixing of these two polymers, so that the API can be reused for 6 months in a single shot. And you can see the diameter is around 4.5 millimeter, and the length is 6 millimeter. The idea was to get a sustainable. So, for example, this implant is releasing API for 6 months. Okay, if I give to you one piece of implant, one formulation, and say that, okay, how much time you need it to characterize the implant. So definitely you will say, okay, I need at least 6 months of release profile, right? So if you don't have 6 months, what you can do? So what we have done is that we have developed a artificial intelligence tool which can give you prediction of 6 months release in 1 minute. So, but before that, we have to understand how the chemistry is there, how the implant is working. So that is quite important to understand the mechanism, real mechanism, because as I mentioned to you, because there is a very less number of research paper available in PLGA, and as thus there is no generic formulation, right? So to before developing such type of artificial tool, you have to have some data, and for that you need to do some experiment, right? We maybe you know that normally the implant is made up of hot melt extrusion. So what is hot melt extrusion? The API and polymer are melt together and mixed together, and then it came out as a strands, and then the strands are cutted by some knife to get this type of implant. And then you can see the time domain for intravenous in vitro release profile. So it is almost about 50 days. So you, for only one piece of sample, you have to invest 50 days of time, okay, which is really time consuming and resourceful. So, and you can see which is here two profiles are there, 502H and 502. So 502 is hydrophobic, and as a result of it, it is releasing API very slowly, okay. And the 502H is hydrophilic. As a result of hydrophilic, the API molecule is getting out of the implant very fast because water is getting affected. Water is getting affinity towards the COH group, and water is penetrating inside the implant. As a result of it, they are making some gateways channels so that the API is going out very easily. And as I mentioned, Ozodex, it's a mixture of two things, right? 502H and 502 polymer. And what we wanted to see is, let's focus on individual implants. For example, implant made up of only one single polymer. And that's why we made two implants with a different chemistry, and try to see how the rate of release is different out of this. Okay. And as a function of it, you can see here, after 20 days in 502, okay, there is some slight release, and here you can see that there is no shift in the pH. But in case of 502, where the API is releasing fast, that means the pH will be quite different. Okay, so it is releasing like it is going down and then up and then down. So this is also one of the characterization methods that you get some idea about pH shift. So you understand from that also what is the mechanism of drug release. So we have this info, right? But this is quite normal routine which everyone is doing in this field in medical device. But what next? उसमें बोलते हैं कि हमने अलग से क्या किया, तो that is really required. What is the mechanism of API and polymer release with limited sets of characterization, what you can do. So we really went out of the trend, out of the box, because normally for release profile HPLC is used, but first time we have used NMR method, NMR instrument, to identify not only the API release but also the polymer release, because the major difference is the API is released because as a function of a polymer degradation. But no, till now there is only single paper, which is our paper, which mentions that how the polymer is degrading or how the matrix is degrading. So you have to understand that. And very interestingly, what we found is that the polymer is releasing first and then the API is releasing. So that means the polymer is degrading because the polymer is a biodegradable polymer. It's degrading and resulting in the channels, creation of channels, which is by which API is getting out. So we have this idea. And then we wanted to understand further how the channels are created. So within the first 7 days, you can see there is some porous structures only in the periphery, but not in the center. Then we thought, okay, let's do more study on this. So we again increase the duration of the study, and when we increase from 7 days to 15 days, the periphery porous structures were totally eliminated, and the internal big cavities are created, which is one of the major force to taking out the API from the implant. And then what we did, this is very, very interesting, guys. Okay. So we have the profile already have 502 and 50H. So 502H and 502. So we try to understand the mechanism and understand out of the understanding of this, we actually predicted the release profile of a combination of these two, which is in the marketed formulation. And you can see it's mentioned in the dash. So this dash thing, so this is virtual predicted, what we predicted without doing any experimental work. And after predicting, we develop our own formulation with a mixture of these two polymers similar to the marketed formulation. And you can see the black line is solid black line is completely overlapping with the predicted one. So that is really a big breakthrough, because now we have the understanding of PLGA degradation, and we can use this as a platform technique to predict the release of API from any implant which is based on the PLGA. And then we made this artificial intelligence tool. You have to fill all the data, that what is the drug to drug ratio to polymer ratio, what is the TPGA, TPSA release profile. So I'm not going in that detail. But if you have if you put this data in upfront without doing any experiment, you will get a profile. And the dotted one is the predicted profile, and the solid line is again the real experimental values, and it is quite really overlapping with each other. So what I understand is that if you identify your problem, you will, and if you have this potential to develop a predicted tool, this tool will be really a significant way ahead forward to reduce the resources and towards the sustainability in formulation development. So this is an overall journey which I have already went through it. I will not go in detail. And the case study 2 is from the nanoparticles, which we have developed with Gallen IQ. It's a big pharma company in Germany, which is mainly doing science in isomalt. So this बचपन में आपने कुछ कैंडीज खाई होगी, तो वो कैंडीज क्या होती है, isomalt. That is actually isomalt, and they have used this isomalt to develop a nano formulations. So which is automatically dispersed, and because the main challenges was that the API was very potent and the therapeutic window was quite close, and that's why they wanted to have some linear profile to get a constant release. So they wanted to have this type of profile. So one is initial rapid release and then trying to maintain the same range. Okay. With the help of nanoparticles or nanoformation. So they use this nano suspension technology with the fluid bed layering, and the Gallen IQ is isomalt, which is acting as a core, and over the core this nano suspensions was actually coated over it with using the fluid bed coater. And this is the same image of the particles, of the nanoparticles. And then they again coated the active layer with the delayed release coating. For example, Udreject is very commonly used as a delayed release polymer. And using these two things, they actually got the two release profiles. One is the immediate release profile, and then a sustained release for a long time. So what I mean is, not only this, okay, what they have done is they wanted to have up to 40% release immediately, and then the 60% release, which should be a sustained release. So very smartly, what they mix, they mixed two samples, 50%, 40% of immediate release, which is without a delayed coating, and the 60% with the delayed coating together in a mix sachet here, and they have given it orally. So the idea is, whatever you want to have a release profile. For example, if a company comes to you and says, okay, I don't want this release profile, I want 20% of immediate release and 80% of delayed release, then you can do the same thing. Like you can add 20% of immediate release formulation and 80% of the delayed release formulation. So that was the idea, that without doing a lot of research work, how smartly you can tackle it to the required output. So you can also think about it like that. If you have if you while you are reading your research paper, if you came across some of the drawbacks or some of the shortcomings, you can try tackle it smartly in this field. So I also like to then give the acknowledgement to Dr. Isha Shah for her help in NMR. Dr. Oliver Leon, which is actually head of the Pharmaceutical Technology Benio, Germany. Dr. Salish Waker and Siddhant Waker for SFA Food and Pharma Ingredients Private Limited in Maharashtra for their collaboration. Yeah, and I would really like to get some questions from you guys, if there is any. Otherwise, I will ask you. Any questions? Come on, guys. Any question, not only for scientific questions, okay, because I understand it's very difficult for you guys to understand immediately all the things, but any questions for your academics, for your professional life, how to approach, if there is any queries or any questions, if you want to understand me that how to approach for this, you can ask me right now. Yes, please. Okay, that's really very good question. AI is, first of all, we have to understand artificial intelligence. Artificial intelligence is mainly developed as a function of your input parameters. Okay, so if you have a sufficient number of input parameters, and you have very good confidence on your data, then on the basis of that, there is some machine learning instruments or machine learning softwares. Some, for example, MATLAB is also there, and other things are also there. So that can be used to take this data and to develop a predictive tool which can suggest you what to move forward. For example, if you have an API, okay, which have a log P of 5, which is very hydrophobic in nature, and you don't know how to move forward with the formulation. Okay, so if you put your parameters of your API, for example, log P value, molecular weight, whatever, the information, then the tool will definitely will suggest you the different type of formulation. For example, for log P value of 5 or 4, it will suggest you emulsions or morphosolid dispersions or implants, whatever you want to target. So, but for that, you have to have very good data of your experiments, and also the data you can get it from literature search. So there is a different variety of literature search. One is your research paper is there, patent is there, and also there are a lot of filings. For example, FDA filing is there, EMA filing is there. So FDA is from USA, EMA is from Europe, MHRA is from UK, and there is also Australian agency is also there, Japan agency is also there. So you have to understand deeply where your data is there, okay, and try to get that authentic data from the literature. Not only rely only on the research paper, because there are a lot of things there, but in the patent and filing, definitely you will get some data. I will just give you a very short example. There was one project with one of the company, and they have mentioned us that they have one formulation, and they want to have an AI tool which can predict the in vivo profile of that formulation. So that in vivo profile you always get it from the patents, and that's very trustable. So when you gather all the information from that source, you put it into AI model, and then you will get a predictive tool from there. Yeah. Any question? Yeah.
Good afternoon, sir. I wanted to ask why PLGL was the best choice as a biodegradable polymer. Instead, there are also choices polyalkyl amino and dextran derivatives and their copolymers.
Yes.
So why the PLGL was the best choice for biodegradable polymer?
Very good question. Yeah, really appreciate that question, because PLG is one and the only biodegradable polymer available in market which is approved by FDA. Apart from that, there is no marketed formulation which have a biodegradable polymer. So for industry, if I am an industry guy, okay, what I don't want science and research. I want my formulation to be in market in as soon as possible, right? And if you want to make a new polymer, biodegradable polymer, then you have to do all the research to explain that the polymer is safe, and that requires a hell lot of clinical trials, which is really very costly. So this is one of the reason that why the companies are not going forward with the other polymers. For example, as you mentioned that there is a copolymer for polycaprolactam and also PLGA. So, and there are also other copolymers, which is nowadays people are developing, few companies are developing, but those companies are not real pharmaceutical company. They are actually excipient company. So once they have the data of the clinical trials, the excipient company, then there will be some formulation which might come in the future. But that's only one of the reasons. And also, as I mentioned, the data or the lack of understanding is already very less for even PLGA. So if you talk about PCL and all other polyamide which you are mentioning, it's even worse. So that's one of the reasons. I think there is one.
Good afternoon, sir.
Yeah.
I am medicine from 5th semester. So basically मुझे ये जानना था कि आपने बोला कि आप एक इंडस्ट्री गए हैं और आपने भी अपनी खुद की इंडस्ट्री चालू की है, तो मुझे ये जानना था कि जैसे हमारे पास कोई आइडिया है और हमें भी खुद का कुछ बिजनेस स्टार्ट करना है, तो उसकी रिगार्डिंग क्या प्रॉब्लम्स आती है और हमें उससे कैसे लड़ना है या फिर हमें उसका कैसे सोल्यूशन निकालना है, तो उसकी रिगार्डिंग अगर आप कुछ बता दो.
Actually, thank you for this question. I was waiting for this question. Look, there are few ideas, right? Depending upon how you are confident on it. For example, I am confident 100% on my product, and I want to make it in market, right? So nowadays, what you can do is, for example, I am also a scientist, and I have really zero or even minus, if you say, minus understanding of all the what CA people are doing. For example, GST filing, company registration, incorporation. There are so plenty hell of a thing there. But if you are confident, then there is also there is one Pradhan Mantri Startup Yojana is there, which is giving you up to 60 lakhs free of cost without any loan interest, and there is also no security required for this. And even if your startup is failed, they are not charging you anything. Okay. One thing is there, and the second thing is all the incubators, which is I'm telling you about high-end incubators. For example, all the IITs, NITs, and other research organizations are also supporting incubators. They are giving you a lot of expertise free of cost to you. Not only actually not free of cost, but because if you are making your startup, for example, your startup name is XYZ Private Limited, so they will take 2 to 3% of your equity. So equity means that 2 to 3% of your company share, and out of this, because of they are getting profit out of this, they will support you for accounts, HR, IT, and all the required documents. So whatever you need. So that in short, what I am saying is, if you are confident on your whatever the idea you have, they will definitely support you. You have to only take care of your science and the context. Again, so this one is how you start your journey. I will say just have confidence on your idea, approach an incubator. They will help you with all setting up all the things, right? The second thing, which is more, I think this is not that important because it is anyway, people will help you out in this case, and it will take around 2 months for you. So within 2 months, from the start to end, you can set up your company. The real game actually starts after two months. After 2 months, because you are new to industry, you have to create your contacts with the companies, right? Because if you want to have collaboration with global pharmaceutical companies, then you have to do interactions, lot of understanding. You have to engage with in conversations with the people. So I think nowadays with the IT sector blooming and LinkedIn and other profiles are like social platforms are already there. So I will say that don't hesitate to interact. Even you don't know, just ask one thing that, sir, ma'am, I need your help or suggestion. I'm 100% sure people will help you out in that case. And they are more than happy, as I mentioned earlier in my talk in the starting, that people are actually looking to India as a next global leader, mainly in pharmaceutical and IT. So, as I mentioned, I have actually mentioned it. I think this Benio Pharmaceuticals in Germany, they upfrontly came to me and, or actually name is me, Alicia. So they said that they want to collaborate, and they upfrontly said that we really want to have some portion of your equity of your company and can give a lot of grant to you and for your ideas. So if you are confident on your idea, first you start your journey with the startup within 2 months, and then start connecting with the market players via LinkedIn or wherever you want to have. So, for example, conferences and LinkedIn, I think there will be the starting points.
Thank you for answering this. Any other questions?
Thank you, sir.
Good morning, sir. I'm Priyanshu Ranjan from semester third. Sir, my question is that you taught in vitro
implants.
in vitro implant that costs around 1,20,000 in other countries.
Yeah.
But how does it reduce to 5,000 in India?
Because, as I mentioned, PLGA is also a very expensive polymer. But the thing is, if you make it in bulk, right? If I am talking about 1 gram of PLG, it will cost you much more. But if you are manufacturing in 1 kilo batch, it will cost less because the costing will be accordingly, right? The second thing is the resources. As I mentioned, outside you have to pay maybe 5 lakh or 10 lakhs of package for a scientist there in a month. Okay, 1 month salary is around 10 lakhs something. But here in India, you don't have to pay that much of healthy amount of money to the people. The second thing is, you also get some, if you want to develop some innovative formulations and some gender formulation in India, it is mostly funded also by the government agencies to develop new formulation or developing a gender formulation for your company, right, for the people of India. And even if you don't get it, the total cost will be actually very less. It will be even 1,000 rupees for one formulation, because here the one formulation is only 1 milligram. Which I showed you. So 1 milligram of polymer and 500 microgram of your API. You can imagine it's really very low. So the major cost is the resources and the time which you spend in developing that formulation. For example, in Innovator, the company developed this formulation for example, 10 years. So the total cost of that formulation is not only that formulation but 10 years of your resources. So that's why it is costing that much. But for generic, you don't need that much of research. It can be in market in a year or a couple of years. So that means that you are already reducing the resources from 10 years to only 1 year. So this will also cut your time point or resources, and also the money. So this is one of the reasons. And even I am betting you that even in 5,000 rupees, if I am saying, you will also get profit, I think, even in that.
Thank you, sir.
Thank you, sir. Another question from this side. Please pass on the microphone.
Hello.
Yeah.
Sir, your company also doing marketing of pharmaceutical product?
No. It's hardcore research and development.
So
I don't believe on marketing. I believe on science.
That's it.
Hello.
Sir, my question is how to open a pharmaceutical company, sir, multinational.
Multinational.
Yes.
That's quite a big challenge.
So drugs and medicines come.
Yes, as I mentioned to you, like start with a startup, okay? And if you get a good collaborators, which I already showed that 1 or 2 months of registering your company, and then a follow up with your how you deal with your clients. So if you have some good links or if you gain good profit,
Sir,
then
where to be registered? Sir,
I think it depends upon where you want to register. For example, there is a incorporation in Madhya Pradesh. So there is also it depends actually where you want to have. So each and every state has their own system where you can incorporate your company. So for the easiest way for you, I think it will be to hire a CA, and he will do all the paperwork for you. I think it will take 1 to 2 months. And once you get to really good, profit, or if your company is starting to grow very fast, then maybe you can try to open it in other countries. Maybe start with low-income countries, and then you can say that your company can be
sir,
MNC.
which license required?
So license depends upon what you want. Okay, so there are multiple type of license. First is manufacturing license or marketing license, as mentioned by him. There is also license for R&D research work, and also in research work there is a different level of licensing. For example, only research or manufacturing in research, or what type of services is there, services, or is there any innovative drug. So there are different type of licensing. It depends upon what you want to start with.
Thank you, sir.
Thank you, sir. I guess this was one of the
Yes, so almost.
everybody must be having in their mind, but we dare to ask. Yeah, and yeah, please.
Good afternoon, sir.
Good afternoon.
Tanmay from B Pharma, third semester.
Okay.
My question is about your second case study
Yeah.
about the delay release of the drug.
Yeah.
Which is the most suitable dosage form, either tablet or you showed the powder sachet form?
It depends upon your API. For example, if your API is BCS class 1 or class 3, then a tablet will be more than enough. Mainly BCS class 1. BCS, there are 4 types of BCS, okay? On the basis of solubility profile. BCS class 1 and class 3 are more soluble in nature. BCS class 2 and class 4 are hydrophobic and not readily bioavailable in nature. So if you talk about BCS class 1, which is high soluble and high permeable, tablet and capsule will be more than enough. You don't need to do really high-end science. But if it is in BCS class 2 or class 4, then you have to develop some novel platforms, like for example, amorphous dispersions, nano suspensions, nanoparticles, or snets, sets, or implants. So then you have to worry about it a little bit. But for BCS class 1, I think tablet capsules is more than enough.
And sir, if someone is interested in research formulations and want to be a scientist in future, what should be the first stepping stone for a bachelor's student to start his or her journey?
The one and most important thing is for a bachelor to get registered to a very good research institute. For example, if you want to focus in India, if you want to pursue your masters in India, I will suggest NIPER. IITs are one of the best institute for research. And then once you get in those big institutes, you will definitely get insights that how to move forward in your PhD or even abroad. Then the most important thing is as soon as possible you get inside to very good research academic career life. In pharmaceuticals, IIT BHU, NIPER Mohali, or other NIPERs and CDRI, they are the best research institutes. And if you want to go abroad as well, it's also possible. As I mentioned, Fulbright scholarship is there, Commonwealth scholarship is there, Endeavour fellowship for Australia is there. There is a lot more for Japan, and Arvind is also in UK. So they are n number of opportunities. You have to search. I think I can give you one website for you guys. There is scholarshippositions.com. So if you register yourself for the newsletter, you will each day you will get notification from them, and they will give you overall all the research possibilities for doing abroad. Okay, they will give you all the details of the scholarships day by day. So you can just register yourself in this scholarshippositions.com, and you can get good idea about that, how, where, in which university, which country you can get affiliated to your master's or PhD program. So this I will suggest, if you want to be a scientist, first to do good science, and good science is only possible in really good research labs, big research lab.
Thank you so much, sir.
Thank you.
Thank you. Any more questions? Yeah. We take this as the last question. Please.
Good afternoon, sir. My question would be, what were the things that helped you in attaining your Commonwealth Fellowship as in your application? Such type of fellowships is very important for financial aids for research students who study in international.
That's quite important, I think, for when you are applying for any research fellowship. Okay, because you have to understand one thing. The research or fellowship providers, for example, Commonwealth or Fulbright or Endeavour, whatever it is, they are not only dedicated to only pharmaceutical, but they are actually considering you as a link between India and the other country. So the link should be in a social or professional platform. So you have to consider that while doing application on this. You have to consider that you also not only give professional value of your work. Okay, for example, I was doing research in cancer, so I was giving importance how the cancer is important for both the countries, and not only for professional wise. I suggested that okay, I can, because I know India very well, and I know it will be a great opportunity for me to also understand the culture and the people of the other country. So if you develop that trust to the examiner, that you are not only interested in science and all other things, but as a social person, you want to do really good things in a social platform, that will be a plus point, I guess. But also, apart from that, you have to qualify IELTS examination for UK, and I think I think there is one more, I think, examination for USA.
Okay, sir. Thank you.
Thank you, sir. Now, and one more question from this side. Anybody want to ask? Yeah, please. Please ask them. This side.
Good afternoon, everyone. My name is Vinod Sir, and I am a first semester B Pharmacy student in Oriental Institute of Pharmacy and Research. So basically, I wrote a research paper on revolutionizing low vision aid for cerebral and macular degeneration. So I filed a patent. So I'm really confused that you said the medical devices. So does it come under the medical device? And I also want to do a startup, but it is a rare disease. Yeah. So I don't think so कि market इतना जाएगा. But still I want to contribute in this because I want to personally pay back to the society what they have provided me, and also about GRE and GNET. I want to say that कि IIT BHU can I just go to Stanford or Harvard somewhere like to do my masters instead of IIT?
See, first of all, there are multiple questions. I will give you answer one by one. Okay. So one is first is your rare disease, right? First question was this. First of all, if you are developing any product out of your research, then it will be, you can use it as a platform technique, not only for that drug, not only for that disease, but for other diseases as well. Okay. The second thing is for rare disease, you can say it as an orphan diseases, right? So for orphan diseases, there is a global platforms which is providing free of cost funding for you, because it's a rare disease, and if you are doing some research in that, they appreciate that effort and they financially supports you. So there is not that big deal for the market. market. Even not only you, big pharma companies like Sandoz, Pfizer, they are also getting funding from government institutes if they are working in orphan drugs. So that is not a problem. Second thing is about GRE, right? So GRE is mainly for a bachelor's and master's degree. Not only even also master's. If you want to go with fellowships, so you don't need GRE. There is some other examinations required for US, and GRE is only for US. But if you want to focus on Europe, there is no such scholarship required. You can directly apply and get the position there. So GRE is not the only thing, first of all, because why I am saying is that GRE is mainly towards English and mathematics, and if you are focusing there, then you will use your time of research, which you want to do, right? So I will suggest you to focus on other streams or other platforms to get into US or UK or wherever you want to go. The second and the last question is IITs versus Stanford, right? I will say IITs are better. Okay, if you are in academics, there are two things. One thing is to gain knowledge, and the other thing is to get exposure. You have to think two things separately. If you want to learn a thing, definitely IITs are much better, much better than Stanford, because I was supervising PhD students and master students there, and the quality of level of education which is required in outside India or mainly in developed countries are not that high as compared to IITs, because here you have a lot of pressure. You have to really go through a lot of screening, examinations, and all of the things. So through that journey, when you qualify different levels of examination, you learn a lot. Okay, but in that case of in Stanford and other things, you don't have to push yourself that hard. So you will learn, but you will learn only to the academics, but not throughout through your journey. But if you talk about your exposure, then definitely Stanford and other universities are much better, because they, as I mentioned, the incubator cells or startup thing are very much there. They are more active towards incubation or innovators, but not in as that good in IITs. But nowadays, what I am seeing is, especially in IIT Kanpur, I visited there. I visited also IIT Bhopal, IIT Indore. Also, I visited. They are now pushing very hard towards new application, new incubator applications, and they are also giving you a lot of opportunities. So overall, in short, I will say if you are keen towards your own goals, if you are more focused by yourself, then you don't need a driving force of Stanford. So you can do it here as well. But in Stanford, because everyone is doing the same thing, everyone is starting his or her own company, then you go with the flow. But here, you have to push yourself upfront. You cannot see other people's, who they are doing, so I will also do this thing. So here it is a little bit difficult, but you will learn a lot here. This is my understanding.
So my next question is how to find research topic, because the macular degeneration in Stargardt was some sort of personal thing to me. But I'm confused now to how to search research topic and how to focus on research, because I have to study my own syllabus and I'm a first semester student. So it's getting hard for me because it's all new. We have maths, we have calculus, pharmaceutical analysis, we have HPLC, chromatography, and all of this is really new. So I don't know how to cope up with the things.
See, a research topic should come from your heart. This is my understanding. For me, cancer was very close to me from my bachelor's. So I knew that I have to focus on cancer. This helped me a lot. I didn't waste a lot of time. But if you are not sure about the research topic, then what you can do, you just go and look at the market size, okay? Which medicine is there? Because if you want to grow in your career, you should be focusing on the topics which is common to the global pharmaceutical company. And in those fields, I think diabetes, weight, obesity, lung cancer, lung diseases, heart diseases, and cancer. So these are the hot topics. They will definitely be there always. So you can focus on out of any one of all these things. So yeah. So you have to either think from your heart or from your brain. So that's this.
Thank you. I hope this would be the last question. And thank you so much, Varun sir, for
Thank you very
spending the
much
valuable
for-
time. I give a round of applause for sir
Oh, thank you.
for sharing his knowledge with us. To do the honors to present moment I will request. Yeah.
Thank you very much. It was really great interacting with you, and I was
So
really
beyond,
expecting for this
beyond
session.
stage, we are going to present you
Okay.
the moment, please. I request Dr. Parul Mehta ma'am, Director LNCP, to come on the dais to present a token of appreciation to sir. I also would request organizing secretaries of this conference, Dr. Gopal Patel sir and Dr. Govind Naik sir, to join for the felicitation.
ये चलेगा क्या ये?
Keep
Yeah,
clapping and appreciating sir for his lot of contribution in the field of pharmacy.
Thank
He
you.
had a long journey, and we may understand that how tough it would be for him to in reaching to such a height in such a young age. Thank you, sir, and many congratulations. We look forward to have more of your knowledge in.
Thank you, ma'am. No, ma'am. It's okay. Thank you. No, it's okay. And if there is any question in your mind, please feel free to email me on this, okay? Not a big problem.
Thank you, thank you.
Thank you. No, it's okay.
Moving ahead with the second keynote session, we have with us our second speaker of the day, Dr. Arvind Singh Chandel, sir. He is a distinguished biomedical scientist with a PhD in biological sciences and over 7 years of international research and teaching experience in biomedical material engineering and pharmaceutical biotechnology. He has extensive expertise in working with diverse material, including polymer, hydrogels, nanoparticle, adhesives, sponges, wound care solution, and antimicrobial agent for cutting-edge biomedical application. Renowned for his originality, productivity, and scientific excellence, Dr. Chandel has authored 36 peer-reviewed scientific publications in high-impact journals. He boasts an impressive H-index of 22, holds 8 patents, has contributed to 20 book chapters, and published 4 articles in scientific magazines and published 2 books. His interdisciplinary expertise and commitment to translating research findings for societal benefit have earned him widespread recognition. Dr. Chandel is the recipient of several prestigious fellowships, including the JSPS Postdoctoral Fellowship, the DBT-sponsored N.K. Bhan Young Research Fellowship, and the Mary Skodwa Curie Fellowship. After completing 2 years of postdoctoral research at the University of Tokyo, he was appointed as an assistant professor, serving 3 years. During this tenure, he specialized in developing modified natural polysaccharide-based bioadhesive for preventing cerebrospinal fluid leakage and artificial oxygen carriers, while leading collaborative projects with multinational companies such as Mokaida Pharmaceuticals and Daikin Private Limited. Currently, Dr. Chandel is Marie Curie postdoctoral fellow at Bernal Institute, University of Limerick, Ireland, where he is continuing to advance biomedical research through innovative projects. A dedicated professional, he remains at the forefront of biomedical innovation, striving to develop transformative solutions for global health challenges. Thank you, sir. We are fortunate to have it here. To do the honors, may I request Dr. Dilip Kumar Tiwari, sir, to come on the stage and present the sapling to sir as a welcome token. Please, sir. Please give them a big round of applause.
Okay. Thank you so much. Thank you so much.
Welcome, Arvind, sir. Now over to you.
Okay, so good afternoon, everyone. So thanks so much for my introduction, and here it's really very grateful moment for me because in front of me my teachers are sitting over here, and it's really grateful moment for me. And in front of teachers, I am also feeling I am a student here. So before starting my presentation, I would like to go through my research journey from schooling. So basically, I am from Singrauli district of MP. I belong to my farmer family. I am belongs to very common family, not so
high-end family. So all of you can correlate with me. I'm not from any extraordinary family. And I joined the Saraswati school. Might you know this school, because it's number of these schools are too much and everywhere in the our state, in over the India. So I completed my schooling from 1 to 12th in Saraswati Sishu Mandir. And after that I joined Barkatullah University, Bhopal, here. And after completing this B Pharma degree, I qualified the GPAT. So after GPAT, it's next to I joined the MS Pharm in Naipar Hajipur. So here, in break of this journey, I will tell you the importance of GPAT. Everyone preparing for GPAT, but I think many people don't know the power of GPAT. So I think India's only one fellowship that can give approx 9 to 10 years fellowship. So through GPAT you can do the masters 2 years. After that you can join CSIR GATE JRF. That is equal to CSIR GATE, CSIR NET. And through that GPAT you can join the RA, CSIR RA. So only one time GPAT can give you 9 to 10 year fellowship. So GPAT, don't underestimate the power of GPAT, because so I think in our education system in India, there is only one fellowship, that GPAT is have that kind of multiple time fellowship chances you can get. If you perform very good in GPAT, so you can avail all of these things. And I get this opportunity through GPAT. So after GPAT, I did the MS form from Naipar Hajipur, and through that GPAT, I joined CSIR GATE JRF in the CSM CRI Bhavnagar, Gujarat. And there I completed my PhD in polymeric biomedical materials. Basically in that the properties of material, how we can design for different drug release, different biomedical devices, about that. And after that, after completing my PhD, I get awarded with JSPS postdoctoral fellowship. That is sponsored through the Japan government. Next, so in Japan, they offer this fellowship twice a year. 120 candidates each terms, means total 240 candidates. They award this fellowship. And here I would like to say, next also sponsor the masters and bachelor fellowship. So in next fellowship, you people can take part in this competition, next fellowship, and at the end I will tell you how you people can do the masters or PhD or postdoc through the next fellowship. Because next fellowship requires one English test, any one of like IELTS or any other English test, and not so high band requirement, like 5 or 6 is 7 is enough for the Japanese country, in Japan country, for the next requirement. After postdoctoral, JSPS postdoctoral fellowship, I was get chance to join that assistant professor in tenure track in the same university, University of Tokyo, and I served there 3 years, and I get chance to supervise the master, bachelor and PhD students there, and I get interacted, I get the chance to teach the students. And after that, after 3 years that I was get awarded with Marie Curie Fellowship, and I went to the Ireland, and currently I am working there. So the main point is that here you people don't underestimate yourself. I am also 10 years ago, 10 to 12 years ago, I was also sitting in those rows. So if you work, and if you really want to grow up in your field, so you need to work hard from the today. Don't wait for your good days. Like if you're thinking, if I will go for master, then I will start reading. No, never comes. That day will never comes. If you focus from today, make contacts, because these fields is not so big. So contact to your seniors, teachers, ask to them, and globally there are many opportunities. You can go foreign for do your master's, PhD, or your internship. So many industries and institutes offers regularly. So you have to be follow up that notifications or ask to your seniors, and you can make your contacts. So that will help. If you think someone will come and help you, no. You have to be show your interest. So here many faculty member was there, and before me, Dr. Varun said that there are many opportunities in globally. So you people should think, and if you are really have interest, you can achieve it. There is no such difficult things in it. So, as Dr. Varun said in one of the his case studies, so there is two type of material he used, hydrophilic and hydrophobic. So that he made for me the easier for today's task. So I will start from here. Amphiphilic. Amphiphilic materials are having two types of components, hydrophilic and hydrophobic in single molecules, single component. So all of you know the plasma membrane. I think we read in 9th, 10th or in biology 11th, 12th. So plasma membranes. The plasma membranes made up by the amphiphilic. So like protein, lipid, protein. So you can see in this first figure. So this is there is two regions, means two parts, the tail of and head. Okay. So here these structures are the inspiring for my research, because these materials through these materials our every cells is made up by such materials which have the both nature, hydrophilic and hydrophobic. So all of you know, I think in B pharmacy students know the micelles, liposomes. These know, all of you know, aware of it. So these are the mainly focused through these type of structure inside the hydrophobic core, outside the hydrophilic, like that. So the amphiphilic materials, my research topic was in my PhD, the amphiphilic materials development. So if we talk about the amphiphilic materials, from contact lens to hard stent, so it has a broad range of applications in biomedical science. So these materials can utilize in drug-eluting stent and many others, implants and other type of stents, balls. Nowadays we're preparing these amphiphilic materials. So I will tell you. So here we can say amphiphilic materials, I told the composition of amphiphilic means hydrophilic and hydrophobic material in one component. So through amphiphilic materials, we can deliver both type of drug. So if hydrophobic drug, we target for hydrophobic drug, the hydrophobic drug will solubilize in hydrophobic region. Okay, and hydrophilic will be solubilized in hydrophilic region, because we know in chemistry the like dissolve like. Okay, so but it should be homogeneous. I think you can imagine through this. So that in the top of that picture, when it's swell in the hydrophobic solvent, the non-polar solvent means hydrophobic solvent, that hydrophobic component will swell, and when it submerges in the hydrophilic means water, then hydrophilic component will swell. So I think everyone is aware of this term gel. So gel is mainly two types. One is hydrogel, that is made up by maybe agarose gel, PEG gels, acrylamide gels, because these components is directly you can solubilize and cross-link it or physically cross-link gel. But amphiphilic co-network gel, that was my PhD topic. So in amphiphilic gel, we combine the hydrophilic component like agarose, acrylamides, and one hydrophobic component like PMMA, PDMA, and or other polycaprolactone. When we make composition, means through any chemical reaction, we join it hydrophilic and hydrophobic, then it becomes one co-network. I will tell you in detail later. That kind of gel, I will tell you here, the amphiphilic co-network gel. So here I would like to tell about the market, because everything's derived from here. If any material don't have like market potential, it have the research on it, it's very difficult, because research need funding. And if we have the market potential, if we have hope of market, then the funds fund by government or private funding bodies, they fund for your research. So if we talk about the amphiphilic materials, these are the main players from BASF to the Reliance Industries in India is the main amphiphilic materials. They are all working with amphiphilic material along with other type of materials. And the market growth rate is very high. It's approx, it will be in 2025, it will be 15% growth rate. So the amphiphilic materials have very high potential in terms of market. Sorry. Okay. Okay. So what are the challenges? Because amphiphilic material is need to be prepared as per our desired properties. Because amphiphilic materials, we are trying to join two different properties of materials like water and oil. Understand? So it's not very easy. There are many challenges like controlled composition. Like if you mix 1 gram of hydrophobic material, 1 gram of hydrophilic material, if you mix and make some formulation, at the end, the composition of your material is not 100%. Some of materials during the process leach out. So such type of problems in this our formulation amphiphilic materials is we have to solve out. So formation of byproduct. If you are going to join two type of materials, there is possibility to form some side reaction product. So that side reaction product, maybe it's harmful, maybe not. But we have to think about it, because everything will go in clinical and it's not easy, because in clinical studies we have to count each and everything, means where it will go, what is the future of the side product, what is the future of the degraded product. So just thinking make gel and we can inject, no. In between that there are huge process in chain of evaluations required. And as Dr. Varun said, FDA approved materials is important. We cannot take any of materials among us. There are many materials, but companies don't allow, because new materials required lot of documentation, lot of clearance for using it. So no one wants to go through that huge long process, because it required too much cost for that. And if you use two type of materials, there is possibility of side reactions, and that side react materials maybe have the cytotoxicity. We have to think about it. Hemocompatibility means if you inject something, it can be may clot your blood, kill your cells. So we have to think about the biocompatibility. I think you all are aware of that. Biocompatibility is very important in terms of if you are going to develop any drug delivery systems. And like we have to tailor our product. Like if you are focusing to sustain release, like for a long time, 1 year, half year, that type of. So you have to, your material should allow to tailor your material for 1 month, 1 day to 1 year. Means not like, okay, if you injected, that material will degrade after 2 years. No, in some case, it's not desirable, because I will tell you in my next slide, some materials required only 7 days or 2 weeks in your body. After that, it need to be degraded. So what I did in my PhD, how I solved these problems, what I told you earlier, to make the amphiphilic materials. So I utilized the nucleophilic substitution reaction for solving these problems. So maybe it will be little bit higher for your knowledge right now, but I think in 12th class you all are aware of this nucleophilic substitution reactions. So in nucleophilic substitution reactions, there is no side product. If you can see, just it joined by the just one the tertiary amine and activated chloride, it go and join together, but there is no side product in these reactions. This reaction not required any special condition. Like if you are going to manufacture, so if your reaction condition is easy, it reduces the cost, because in manufacturing stage we need to each and every step we have to think about the cost of your final material. So these reactions did not require any special conditions. And these reactions is very easy in the room temperature. Like if you mix material and inject somewhere in body, it can cross-link inside your body. So this reaction does not generate any heat inside the body. If any reaction generates heat, that cannot be used for the clinical purpose, because heat can kill your cells. So we have to choose very carefully materials in your reactions. I will tell later. So first of all, this click type of reaction is click type of reaction means there is no side product formation in the nucleophilic substitution reactions. There is no leaching product, because just it go and join, no side product formation. So we have to not think about the side product biocompatibility, cytotoxicity about it. In fast gelation, so like if you are going to deliver some drug inside the body, so you mix with your component and inject it. So we need fast gelation, not like if it required 1 day or 2 hours, it's not desirable, because if it take long time, it can be released out from your site of injection. So this reaction can possible in settings of US FDA approved materials. I have used the polycaprolactones in other polysaccharide based materials. Its material is biocompatible and easy to tailor. I told you the material can be tailored according to your applications. If you need fast degradation, you can tailor it, reduce the cross-linking density, your material will degrade faster. If you want long time of degradation time, like 6 months, 1 year, you can increase the cross-linking density, increase the molecular weight of the precursor molecules. You can enhance the biodegradability of your materials. Oh, sorry. And this material can tailor if you go in other applications, like where it required mechanical strength of material, basically in the bone regeneration area. So there required the material mechanical properties, especially in other area also. So you can tune the mechanical strength as well. It can be inject or as I told you, it can be delivered through injections. It can help the regeneration of your blood vessels inside. So if imagine if you have wound and you deliver your hydrogel over it in wound. So first of all, it should be protect your wound. Second one is regenerate, means development of new arteries, new veins through that hydrogels. So your hydrogels should support the regeneration of new arteries or new veins. And so such materials are mainly present in the some material in market. For wound care, as I told, in wound care, many materials are already available in similar type of materials, as you can see. So here I will give one example what I developed in my PhD. So here we have taken the agarose. So I think this term you know, agarose. So agarose is polysaccharide, is obtained from seaweed. And we modify the agarose with the tertiary amine. And that tertiary amines will help in the further cross-linking. And we have developed here the cross-linker, the tri-block cross-linkers. And it has the ester bond. Here I will tell you, this is ester bond. This ester bond will help to degradation, to control the degradation of final material. So it designed according to that. And when it mix, it form this kind of film, transparent film. And this film can load the drug, and I will show you. We load and release the drug. And these materials, we actually, because it was in research, so we have to not satisfy with one composition. We have to change the composition from lower end to higher end, and we have seen where it will be very best composition. So we found that this, when we mix two components like agarose polycaprolactone, means cross-linker and agarose amine. So in the third column, you can see the salt fraction. Salt fraction is that like if you mix A component and B component and it's cross-linked, and when it wash it, because before using we can wash it. So during washing, that percent of material is washed out. So actually salt fraction, that is too high salt fraction, because if some component is washed out, that will change your material's property. Like if you have a special requirement, but you cannot achieve through it, that if it was out, so final composition, as I told you, final composition estimation is very tough. And if salt fraction is high, that can change the strength of material, because some part is washed out, so we don't know what component, like component A, component B, how much is inside that. So we don't need high salt fraction value in our product. If it is high, it cannot be used further, because we cannot tune it as per our applications. So as I told you, so I use two type of material, amphiphilic, like hydrophilic and hydrophobic materials. So if you, how can you check your material is very properly mixed? Like it can be one side hydrophilic, one side hydrophobic. So we have to evaluate it for the mixing of material. It should be homogeneously mixed. So you can do the AFM analysis. I have performed here AFM. This is a spectroscopic technique. You can imaging through it. Here you can see the first image in the middle row. This is very homogeneous. You can see this color distribution of is very homogeneous. But in the bottom side, it's not homogeneous. So you can, through this techniques, we can evaluate the material's homogeneity. And finally, we need homogeneous material. And here the AFM, and you can also do some color. If your material is some charge, so you can absorb some color. If color is homogeneous, here I absorb the rose Bengal dye, and you can see this dye is homogeneously distributed on this film, so that also support the result of AFM. And also next we perform the degradation, as I told. So our materials degrade 25% in 60 days, and different compositions degradability was different, because as I told, the salt fraction was high. So I will tell you how to reduce the salt fraction in next slide. In this material, I loaded the drug 5-fluorouracil, anti-cancer drug. This drug is hydrophilic. So here 5-fluorouracil was used as model drug for drug release experiment. And we performed here the 300 hours drug performance, release performance, and hydrophobic drug, prednisolone acetate is hydrophobic drug, and here loaded for the hydrophobic drug models. And also this drug was released approx 50% in 300 hours. And biocompatibility, we tested the biocompatibility, hemocompatibility using the cell lines. So our materials should be biocompatible. So for performing, for checking that your material is biocompatible or not, we have to grow the cells on your materials. If your cells grows, this is in vitro test. So if cells grow very well, is proliferate is normally. So generally this is a first like primary experiment for checking the biocompatibility. Then you can go for the in vivo, like use some model animals in the lab. You can implant your material, inject your material, and leave it there and take the tissue from there and do the staining and see the fibroblast and other cells migration of there. So that is the further steps of that. So biocompatibility is very important factor. And this work was published in the American Chemical Society's paper. So in this part, I will tell you how to reduce the salt fraction. Means we are going to use some material that can promote the mixing of two different type of materials, like hydrophilic materials that are soluble in water, hydrophobic material that is soluble in polar and non-polar solvent like oil or some other non-polar solvent. But if you mix directly, it will be not soluble. We need some material that I will tell you compatibilizer. Here these are two compatibilizer I have used. When I use two compatibilizer, it mix well. Here you can see the agarose and polycaprolactone is mixed with this compatibilizer PDMA. This PDMA, when it's here you can see agarose and polycaprolactone solution is not transparent, but when you add this PDMA, this become transparent. This transparency is indicated that this system is homogeneous now. So miscibility, it's enhanced the two different natures materials miscibility to each other. So this PDMA block NIPAM, these are used as compatibilizer. So I will not go in detail, but that I told you that agarose amine and polycaprolactone, means chloride terminated polycaprolactone, that have the high salt fraction, means during washing some part was washed out, but when we used this compatibilizer, these materials salt fraction was reduced. You can see that salt fraction value from 40%, earlier it was 40%, that reduced to the 2.1% when that compatibilizer mixed with different system. So we solve that high salt fraction problem through the design of compatibilizer. So after using that compatibilizer, we make that several films and we evaluate that through the different techniques. So like DSC, I think in analysis subject you will be read in, I think in third year or fourth year, DSC and DMA kind of, these techniques I think is there. And these are the images of our final product after using the compatibilizer. And this compatibilizer was having the it's incorporate two other properties in our previous materials. Means it have the pH and temperature sensitive. So this material can be change the release rate through the change the pH and change the temperature. So like you can see here, the materials swelling of this material, it changed through the changing the temperature from 37 to 17 degrees Celsius. Because polynipalm, here we use polynipalm that is temperature sensitive, and PDMA is pH sensitive material. So after incorporating that two materials, it changed the property of our final materials. And also we check the release rate of this through that, and it also enhanced. Before that, it was very high release and faster release, but after incorporating that material, we improve our materials. Okay, and this work was published in the polymer journals. So next is gel. So gel, when I told you gel materials, when we prepare the gel through the hydrophilic materials, like that is soluble in water. But here I am telling to the challenges of gel, because some traditional gels is good for certain materials, but not good for other type of material like hydrophobic drug. So mainly I focused here to deliver the hydrophobic drug through the gel system. So there is many problems in hydrogel system, injectability, because if your material is hydrogel is not injectable, so it in clinical applications that limits your application in clinical settings. Biocompatibility means your material should be biocompatible. And mechanical strength, because some gels swell a lot and lose their mechanical strength that cannot use in several other like bone regeneration or other fields, because they need certain mechanical strength. And hydrophobic drug loading is one of the most challenging tasks in hydrogel systems, and responsiveness and tunable degradability, because we don't need long time or short time. We have to design our materials as per our applications. So here I design the material that have the gelation time is 2 to 4 minutes. So when we mix this PDMA block napalm and PEG materials, it cross-link through this through the amphiphilic nucleophilic substitution reaction and form here the positive charge, and that positive charge will help in the applications. So, and this material is, as I told you, have the temperature responsive and pH responsive. You can see the hydrogel have the at 15 degrees Celsius is transparent, but at 37 degrees Celsius at body temperature it's become turbid, because napalm have the property it become hydrophobic, behave hydrophobic at the body temperature above the LCST temperature at 37 degrees Celsius. Sorry. Okay. So when we see gel in the electron microscope, SEM, so gel have the porous structure. So if you grow the cells, so these pores provide the space to grow the cells and transfer the nutrition from these channels, and it support the proliferate the cells and allow grow the cells, and it provide the healing at the application site. Sorry. And also these gels show the have the positive charge. As I told you, due to the positive charge, this gel can coagulate your blood. So like if some wound is there, you can apply this gel, because positive charge will help the attract your RBC, red blood cells. Red blood cells have that negative charge because the membrane of red blood cells is negatively charged, and attract through that this charge, positive charge, and it helps the hemostasis applications. So this gel can also help in the hemostasis. And this work was published in RSC journals. And as I told you, hydrophobic drug loading. So here dextran, one of the polysaccharide which I used for this hydrophobic drug loading, and we modify the dextran through this PDMA, through this ATRP reactions, and is modified with alkyl amine. So here this material is amphiphilic because this is hydrophobic component, this is hydrophilic component. Here you can see clearly the amphiphilic, how it's work, amphiphilic material, and as I denoted here, dextran graft QPDMA as this whole material. This is amphiphilic material, but here you can see this is hydrophilic. Here up to here it's hydrophilic materials. and when it mixes with polyethylene glycol, the chloride terminated polyethylene glycol, it reacts and that form the hydrogel in 2 to 4 minutes. It takes 2 to 4 minutes, and this gelation time also can be enhanced through the concentration and temperature. So this is video of this gelation, but I think it takes a long time. So how it will be work in the lab? So you can see when you mix these gels, two components, and it forms the gel very quickly. I think if it so these two components, if we mix to each other, it forms the gel. In this solution, before gelation, there is 2 to 4 minutes, you can inject through it the needles. So it allows that time, before gelation time, it gives time for your gelation. You just mix it and it forms the gel. After mixing, you can